Okay, well, welcome everybody. Again, thank you all for coming, and thank you in particular for the Mecklenburg County beekeepers for hosting us this evening. As you all know, I'm not in Mecklenburg County, but they are graciously hosting us tonight on their regular scheduled monthly meeting. And so this way I'm able to not only speak directly to the entire Mecklenburg County beekeepers, but also the Onslow County beekeepers are joining us, as well as the beekeepers of the Albemarle, as well as several dozen other individuals who are joining us, and so welcome to you all. Particularly want to thank Bonnie Jones, Tom Davidson, and Jerry Mack for doing test runs and for coordinating this and for inviting me probably about over a year ago now that we've set this up. And so as we got closer to the date, we decided that it would be very timely to talk about this particular issue of the NCSU Swarm Collective. So this is a new extension initiative that I've been thinking about for many, many years, almost 10 years now. But I've finally decided to pull the trigger this year because I think the timing is right, and I think there's some very, very good opportunities where we can learn from each other as beekeepers. And so this is one example of some of the citizen science projects that are going on in beekeeping. And so here in North Carolina, we'd really like to harness this, particularly because we have such a vibrant and large beekeeping community in the state that I think we can really make some very impactful outcomes from something like this. So before we get started, I just want to make a couple brief announcements concerning issues relating to apiculture and beekeeping in the state. First and foremost, of course, is the upcoming summer meetings, particularly that of the NCSBA. And so that's going to be in Norks Wilkesboro next month, July 10th through 12th. Hopefully all of you are going to be there. It's usually a really fantastic event. 500 or so beekeepers learning all sorts of different aspects about bees and beekeeping. If you've never been to an NCSBA state meeting, I definitely encourage you to go. You will not forget it, and you will be glad that you went. Similarly, the Eastern Apicultural Society Summer Conference is a regional conference all the way from Maine down to Florida and everything in between. So it's, again, about 500-600 beekeepers this year will be convening in Richmond, Kentucky, the very end of July and beginning of August, where, again, they're going to have a really fantastic short course and then conference that deals with all different aspects of bees and beekeeping. So you can just see both of those websites to attend and how to register and how to attend those conferences. I was also asked to say that we're going to have another webinar just like this, using this same Collaborate format, specifically devoted to the North Carolina Master Beekeeper Program. And this is a way for the different county chapters to liaise directly with Dr. John Ambrose, who is running the Master Beekeeper Program on behalf of the NCSBA. And that is going to be Monday, July 7th is the day that, or the evening that that's going to be. It's going to be from 7 o'clock to 8:30 that evening. So much like this, there'll be a question and answer session or some ability to coordinate with Dr. Ambrose about the Master Beekeeper Program, because there's been a lot of questions about that program that you have for him, and so that will be a good venue for you to interface with him. Okay, so the citizen science movement is something that I'm sure you've heard a lot about in all sorts of different aspects. I think with interconnectivity, the internet, web design, maps, all these different tools that we now have available to us has really facilitated doing things like this, where people can come together and do projects that are of scientific merit. And this is something that we conducted. We had a little taste of this way back in 2005, which seems like forever ago, but it also seems like yesterday, when we launched a project funded by the Golden Leaf, where we provided pairs of hives to new beekeepers. And this was a way to really help generate a kind of a vigor among the beekeeping community here in North Carolina. And by doing so, we were able to give away 500 hives. So that was 250 new beekeepers that were part of this Golden Leaf program. And when we advertised for it, there was something like 2,800 applicants that actually vied to get these pairs of hives. And so there was obviously not a very good likelihood that any one individual would be taking part in it, and so many of those beekeepers just went ahead and bought hives themselves anyway, and so it really helped to grow and bolster the local beekeeping community here in North Carolina. And one part of that for those actual participants is that they were given pairs of hives. And they were supposed to be all Russian bees, these new genetic stock that are tolerant to Varroa mites. But we weren't able to secure a sufficient number of Russian queens. And so what we ended up doing was providing one Russian queen in the packages of bees and hives and another Italian queen. And so as we were going through this, we realized that this was actually a very unique opportunity to be able to do a comparison of these pairs of hives that were given to these 250 new beekeepers, and we simply asked them to measure the number of mites within the colonies at the end of the summer. And so in doing that, they went through and they monitored and measured their mites, and they sent those data back to us, and then we statistically compared the colonies headed by the Italian queens and the colonies headed by the Russian queens, and we did indeed find a statistically significant, a scientifically meaningful decrease in the number of mites of the Russian bees. And what was really remarkable about this is that these participants were all over the state. They weren't in one region, they were randomly scattered all throughout the state. And so it really shows this is a real world example of citizen science where they collected the data and they really showed how robust these different genetic stocks can be in helping Varroa mites. And so this was published in American Bee Journal and really showed the power of citizen science. And ever since then, that really got me thinking about what else can we do along these lines. Since then, of course, we've also been very much involved in the Bee Informed Partnership, or the BIP. And this has been, again, it is a nationwide initiative that involves dozens of different collaborative institutions and working together and partnering, again, with beekeepers. And so every year there are these surveys that go out asking beekeepers to give to record what they've done in the past year and how well their colonies have done. And every April, in fact, there's the annual winter loss survey where they try and measure how many bee colonies died off during the winter. And this is something that the federal government doesn't do, that there really isn't an infrastructure, but this has been doing a lot of that on behalf of the apiculture community nationwide, and in fact has been providing that really important data about whether colonies are dying off at alarming rates or not. In doing so, there's also a management survey asking beekeepers much more detailed questions about different management practices that they do and the effects it has on their survival, on the colony's survival. And so this is just a graph of just one of hundreds of different data outputs from the Bee Informed Partnership, looking at different mite control products, for example, and the likelihood that their colonies die. And so it's a kind of a crude measure, but nonetheless it's very, very informative to partner with beekeepers and for all of us to share our information so that we can learn from each other. And that's the philosophy of the Bee Informed Partnership. However, the overall goal of that is to just look at colony mortality as a measure and nothing more finite. And so with those examples of citizen science that our NC State Apiculture program has been involved in, I thought it might be time to launch what we're calling the Swarm Collective. Now, this is not a project that has anything to do with the swarms. I'm just using the analogy of kind of the mass chaos of tens of thousands of bees flying around, but they end up actually making sense, making decisions, and being able to accomplish a task that they can't do individually, but that together they are able to. So in essence, it's just a citizen science initiative where we want to pool common data among different beekeepers and test relevant hypotheses. And so in doing so, what I'd like to do is every year have an experiment that we will collectively do. And it's a challenge to all beekeepers out there to participate in this Swarm Collective and try and work together collaboratively to try and test these different hypotheses. And every year, hopefully, we're going to have different hypotheses that we're going to test. So in doing so, I want to set up different experiments that are going to be relatively simple. We can't make them too complex. We can't have, that the requirement is that you have to test 100 hives or something like that. That it's going to be fairly modest requirements of the number of hives and the inputs that it's going to take to conduct this study. So I think if we have a relatively low bar, that will increase the participation, or at least the ability for most beekeepers across the state and nation to participate in this. And so by pooling all of our data together, the idea is that, we can learn from our own management practices, and that helps us become better beekeepers. But nobody is smarter than everybody, in the sense that we can learn so much more if other people are doing the exact same thing, and how those different results can actually manifest themselves, and whether we were just lucky and on average this, whatever it is doesn't actually work, or whether we were just unlucky and for the most part these things actually do work. So we really need to kind of come together and share those data, because not everybody's beekeeping experience or outcomes are the same, even by doing the same thing. And so there's this website here that's off of our Entomology Cooperative Extension webpage, where I will be posting the Swarm Collective annual explanation and further instructions about how to conduct that year's experiment. And so the idea is that in the spring or in the winter, we will develop this, and we will post it. We will disseminate the call for beekeepers to participate in it. Over the active season, we will do the measurement. We will conduct those studies together, and then we will pool our data together, and then we will analyze them in the fall, and then publish them either online or in a beekeeping periodical, so that we can all learn what the final results were. And so keep posted onto this website year to year. We hope to have different challenges every single year. Now for the current year, 2014, I thought that we would probably, to start off as easy as possible, and to learn from our experiences from the Golden Leaf Project and from the Bee Informed Partnership, that we would focus on Varroa mites. And as we all know, Varroa mites are among a top priority, if not the top priority, for keeping our colonies healthy, right? And so our best management practices that we all abide by calls for an active monitoring and management protocol for Varroa mites. we really do need to help the bees out for Varroa because they are not a native parasite to our honeybees. And so it's really good for us to help the bees out, and there are many different things that beekeepers can do to try and control Varroa, or at least help the bees control Varroa. And so the idea here is to pool our data so we can see which of those mite control practices are more effective than others. And so again, if you're a beekeeper, you should be measuring your Varroa levels within your colonies and hopefully doing something about them if they get out of control. And therefore, you should be taking those notes. You should be doing those measurements anyway. So why not share those numbers in a systematic way so that we can all learn from each other, okay? So the objective here, again, to keep it fairly simple so that pretty much any beekeeper can participate, is that we're going to measure the levels of Varroa mites within pairs of colonies. So you have to have, at minimum, 2 colonies. And then we're going to measure them several times over the course of the season, and then measure them before and after some sort of mite control intervention. And that is entirely of your choice. And then we will measure before and after, and then see what the efficacy or how good that control measure functions. Okay, so as I said, we're going to do pairs of hives, and what the kind of the basal assumption of such a comparison is that everything needs to be exactly the same, or at least as close to exactly the same as you can. That is, you want to standardize everything except for the one thing that you want to test. In this case, it's whatever mite control strategy that you're evoking, okay? So your pairs of hives, you want those 2 hives to be as close and similar to each other as possible. You don't want one to be a 5-frame nuke and another one to be, you know, 2 double deeps and 5 supers on it, okay? You want them to be similar size, similar strength, similar health, everything the same pretty much, okay? So you want them to be in the same location. You don't want one to be in a good location where there's a good nectar flow on, and then another in a poor location where they're going to be stressed out, right? You want them to be close to each other, right in the same location, same size, same everything, as close as you can. Now, in doing that, now you have 2 hives that are pretty much one and the same, that they're not really different from each other. And then you're going to compare them to each other, right? So one hive will be what we in the scientific parlance is called a negative control. And that's in essence where you don't do anything. And in this case, with respect to Varroa mites, you just leave it alone and you don't really manipulate it in any other way. Then the other hive is called the positive control, which is where you are actually implementing your mite control strategy. And it's, again, whatever you do in order to help your bees control the Varroa mites. It can be a pesticide or a miticide. It can be one of the biopesticides. It can be a mechanical approach like drone brood trapping or sugar dusting. Or it can even be a genetic stock, if you wish. So there's lots of different options out there. We've talked in these webinars before about different mite control strategies. We have courses online that talk about these different mite control strategies. So whichever one of those that you elect, you do it to that one hive, but not the control hive, okay? So the idea then is that we're going to measure the mite levels in both colonies twice before and then twice after you implement that control strategy in that one hive, right? So we have a before and after of the one that you don't do anything to, and then the one that you do something to. And the idea here is that following standard kind of mite levels, they increase throughout the summer. So the one negative control hive, those mite numbers are continually going to climb. Whereas the ones in the treatment colony, they're going to go up and then they're hopefully going to be knocked down the mite levels are going to go back down after you implement that control strategy, right? And then at the end of the experiment, if the control hive gets too high, then hopefully you can implement some sort of mite control strategy for it so that it doesn't experience too many problems going into the winter. But after the experiment is done, that's very, very important, right? So the control hive, you don't want to implement any mite control strategy during the experiment itself. And we'll get to the timing of that in a second here. Okay, so what you're going to need, again, fairly minimal inputs that is going to be required of you, and again, things that you should already be doing. So a minimum of 2 hives, and if you want, you could do multiple pairs if you want to do it, but do it in pairs. But a minimum of 2 hives, again, as equal and as standardized as you can possibly make them. You're going to need 4 sticky boards. That is the adhesive sheets that go on the bottom board of the hive per colony. So you're going to need 8 sticky boards for each pair of colonies. Again, measuring twice before and twice after. You're going to need a sugar shake or a sugar roll jar or apparatus, which again we'll talk about here in a moment. And again, most of you should or already do have one. And then if you have trouble identifying Varroa mites on sticky boards or coming out of a sugar shake, they're visible to the naked eye, but some naked eyes are better than others. And so if you need a magnifying glass in order to really verify that what you're seeing are Varroa mites, then you'll need one of those as well. Okay, so for the software, you'll also need whatever mite control strategy and product that you might be using. Again, if you're using Apilife VAR, ApiGuard, formic acid, any of these different biopesticides or products that are out there, you will need that. Again, if you rely mostly on mechanical intervention like drone brood removal or powdered sugar dusting of the entire colony, you'll need that as well. So that kind of is where you as the beekeeper have choice about what you're going to implement here. Of course, you're going to need powdered sugar and other things for the sugar shake measurements of the colonies to measure the mites, right? And then you're going to need a notebook or a data sheet that can be downloaded from that website that I had shown before, as well as other instructions about this entire process, or just some sort of notebook, your notebook that you have, your apiary notebook, where you can write these things down as you go through the course of the season, because you're probably not going to be able to remember them. The handout, the downloadable PDF that we have on our website, it has a nice table there where you're able to write all the information down that will be sent into us that we'll be able to analyze. And so it's a nice, easy way to record all of these numbers. Okay, so the reason why we're asking and why we think it's a good idea to measure Varroa mites in these 2 ways is that there are pros and cons of different ways of measuring things, right? So there are trade-offs with accuracy, with how much time it takes, with the expense, and what actually is being measured, okay? So really we want data, we want information that's going to be robust, it's going to be repeatable, it's just going to be reliable for us so that we know when we analyze it that it's going to be meaningful, right? So the sticky boards are really good because they're passive. All you have to do is shove them into the colony. It's fairly quick. Unless you have a lot of mites and it's kind of hard to count them all. And it gives the overall load, that is the total number, or an estimate of the total number of mites within the colony. The downside to the sticky board is that bigger colonies obviously tend to have more mites. And so the total number can be higher from one beekeeper to another or from one hive to another, just simply because the hives are bigger and the colonies are bigger, not because there are more mites, if that makes sense, right? So that's kind of the downside of the sticky board. The sugar shake is the opposite. Right, so rather than the mite load, it's actually giving the mite prevalence, that is the percentage of bees that have mites on them, okay? So it's very good in the sense that it's providing kind of a more percentage number that's the same or equivalent between smaller colonies and larger colonies. It's non-destructive in that it doesn't actually kill the bees, and it's independent of colony size, but it can be highly variable. That you can do one sugar shake on a colony and you go back into that same colony and do a second sugar shake and you actually find different mite levels, okay? So it's, it can be subject to some stochasticity there depending on just your random luck as you go into a colony, right? So it's helpful to have both of those measures. At least that's what our previous experiences found. And so I think that's probably a good thing for beekeepers to be getting into the habit of, not just relying on sticky boards or not just relying on sugar shake, but trying to get that perspective from both of those vantage points. And that can make you more informed and enable you to make kind of better management decisions about your bees. Okay, so again, for those of you that are unfamiliar with sticky boards or with sugar shakes, there are videos on the website that go through these. I did a trial run here to see if I could show them through the Collaborate, and the videos weren't working. So just go to those websites, and they're, you know, kind of 2 to 5 minute videos that actually show what to do and how to measure mites using sticky boards or sugar shakes. But for the sticky board, pretty much you can buy these things commercially, or you can make them yourself just with some white card and putting some adhesive, some Vaseline, some pan cooking oil, just a thin layer, so that as the mites fall down from the brood nest, they'll adhere to the sticky board sitting on the bottom board. Okay, it's helpful to have a wire screen or rubber mat on top so that the bees themselves don't get stuck, but only the Varroa mites do. And you just simply slide that into the entrance and you leave it there for 24 hours. If you leave it in there kind of for a shorter period of time, then you might not get as many mites and it might not be as accurate a sampling. The standard time is about 24 hours, but if we leave it in there for 72 hours, it'll really give us a good idea of kind of the total mite load within the colonies. And again, you're going to want to do this twice before and twice after whatever mite control strategy you decide to implement. So this is a sticky board that I would say was either from a dying colony or one that was already dead and it just didn't know it. Each one of these little brown oval dots on the sticky board, which often has some sort of grid or outline of a grid within it, those are all Varroa mites. Here's a slightly blown up version so you can see them. They're ovate. They're reddish brown. They're very consistent. Sometimes they can be chewed a little bit, or some legs or a little section of it missing, but in general, this is what the Varroa mites look like. So what you're going to want to do is you're going to want to count the total number of mites on the sticky board. Now, I do not envy anybody who pulls out a sticky board from a colony like this, because you might not be able to count that high. I certainly can't. But hopefully most of your colonies are not going to look anything like this. But it's still important to go through and systematically, square by square, count the total number of mites on that sticky board. Now the sugar shake, again, the sticky board is good because you get a lot of mites sometimes, and it gives the total mite load within the colony. But the sugar shake is something that can be done a little more quickly in some ways, and it gives you a percentage, that is the mite prevalence, right? And so what you do is you fashion some sort of jar, and again, there's a video on this about how to do this, and we also have beekeeping notes that can walk you through exactly how to do this. And again, hopefully most of you are already doing this. This is something that every good beekeeper should be doing. And you get a mason jar or an old peanut butter jar, and you take the lid and you cut out the lid and you fashion it with some wire mesh. And then what you do is you go into the brood nest and you hold hopefully a frame without the queen on it, and you take the jar and you kind of gently scrape the bees, tilting it towards you like this, and as you scrape the bees into it, they'll fall into the jar. And what you want to do is you want to get about 200 or so bees into that jar. Now, what a lot of people do, what we do at the Bee Lab, is that we have we take some old bees, some frozen bees, and we count them out to 200. We put them in the jar, and then we draw a line as to the level at which those 200 adult bees really should be at the bottom of that jar, so that we can just guesstimate it by volume. Okay, and so you get those 200 bees into that jar, and then you quickly screw on the lid. And then you, through the wire mesh, you dump the powdered sugar on top of the bees to coat them completely. Not so that they're drowning in the sugar, but 1 or 2 tablespoons of powdered sugar so that they're completely coated in the powdered sugar. And what happens is that the mites that are clinging onto the backs of the bees, they lose their grip because the powder gets onto their leg parts, so they can't hold on very well. And so then what you do is you invert the jar, and preferably on a white piece of paper, you then kind of gently, well, you kind of have to do it fairly vigorously, shake the excess powdered sugar out of the jar. Now the bees won't come out because of the wire mesh, but the wire mesh is big enough to allow the mites to fall out as well. And so what you'll see then are these mites covered in powdered sugar that will fall out, but the bees won't. And so that's a way to separate the mites from the bees. And then you just simply count the number of mites that fall onto the piece of paper, and then the number of bees, or an estimate of the number of bees in the jar. Some people do only 100 bees. Some people do 300 bees. That's up to you. But whatever it is, you just simply take the number of mites and divide it by the number of bees, and you have a percentage. Lisa, your hand's up? What level was the frozen bees? So as we did that kind of calibration, right, we use old peanut butter jars, old Jiffy jars, and they are about 1.5 inches off the bottom for 200 bees. Now again, that's going to depend on the volume and everything else of the specific jar that you're using. So you can use a Jiffy jar if you want and do it at about 1.5 inches, or just kind of guesstimate it yourself. Again, there's going to be some error in this, but you want to be consistent, and you want to be as accurate as you can. You can just eyeball them, as you know, that's about 200 bees. You can try and do that, but usually, if you can, try and calibrate it with some known quantity of bees, if at all possible. And again, we have literature and videos kind of explaining this online. So this is Don Hopkins, our excellent state apiarist, doing that, and this is also this is Bill right here as well. They're demonstrating this and just shaking it out right on the inner cover right here, and showing the different mites that fall out from the sugar shake, and you can see them right then and there, right in the apiaries. But again, don't forget to write down the number of mites that fall out of the sugar shake and the number of bees in the jar so that you can get that percentage. Okay, so again, the idea here is to compare treatments, and I won't, again, go into all the different treatments that are out there. We have literature on this. See our beekeeping note 2.03, and there are many things also that are not discussed in there. But it's actually fairly surprising how few studies have gone into kind of side-by-side testing these different things. Now, again, you're not comparing one treatment to another. You're comparing doing nothing to using something, right? So, but that pairwise comparison is a very powerful approach to really see the efficacy of these different mite control strategies. And so the way I'd like to see it, or to conceptualize it, is that you have different types of mite control options that vary in their degree of manipulation. So some things are very passive, like screen bottom boards and using mite-tolerant stocks and other things like that. Other things you really have to do a lot of work in order to implement that strategy, right? So there's kind of a continuum of different degrees of manipulation. And then how well they work, the efficacy of these different things. Some of them don't work very well. Some of them work extremely well. And so you kind of have all of these different trade-offs, right, of things that work and things that don't. And so this is what we hope to accumulate data on, that if enough of us use the same different mite control strategies, that we'll be able to pool our results and compare our results and really see on average what is working and what isn't. And then we'll be able to share that back with each other anonymously, of course, so that we can really say, oh, okay, you know, what I've been doing really seems to work, and others seem to have that same experience. Or, you know, what I'm working on average doesn't really seem to work all that well. I might entertain, you know, some of these other options. So that's what we're going to be doing with this. And again, it's going to be doing it measuring the mite levels. Once in July, really pretty much any time that you want, once in August, before you actually do that manipulation of your treatment colony, okay? And then a second time in late August, after you've implemented that mite control strategy, okay? And then one last time in September, again, hopefully giving you enough time to treat the control colony if those mite levels are kind of alarmingly high, all right? So really, there's just going to be 4 sticky board counts and 4 sugar shake counts for each colony at each of these time points. So it's only really 20 data points that you have to collect. And again, the good beekeeper is already doing this. The best management practices during this time is when the colony populations are starting to level off and even starting to dwindle a little bit, but the mite levels are really ramping up. So this is the period of time probably when you would need to take some sort of action anyway. And so really the only difference is that you're writing these down and sending those data to us, and for one of your colonies in your pair, you're purposely not doing anything, in essence, to see how bad the mite levels could get if you didn't do anything, okay? Now, in doing so, it's also going to be helpful to collect some other non-personal information that also have some bearing on the mite levels within the colony, just so that we can kind of standardize among different beekeepers for different things, like location, and of course what mite control strategy that you decided to implement, and importantly, how those colonies were established earlier that year, whether it overwintered, whether you started as a nuke or started as a package, because those things kind of talk about the maturity of the mite population as well as the bee population. So I think those things are something that need to be included. Also, whether or not you use screen bottom boards. Again, unless you're doing it as your mite control controlled treatment, you want to have them both hives having screen bottom boards or both hives having solid bottom boards. right? Otherwise, you're going to want to have your control colony with the solid bottom board and your treatment colony with your screen bottom board and nothing else different, okay? I can tell you that while it is helpful to have them, that is not going to probably be a sufficient means of keeping your Varroa levels in check, just in and of itself. But it's still something that has an effect on mite populations, so we need to account for that. Similarly, the type of stock that you used. What we want you to do is use the same genetic stock for your 2 hives in each pair. Again, unless it is the treatment that you're using to try and assuage the Varroa mite levels. So if you're testing formic acid, for example, then your control colony gets nothing, your treatment colony gets formic acid, but both are Italian bees or both are Russian stock. It's the same stock, right? So the only thing that's different is your treatment, okay? So we're going to need for you to report the stock type that you have within your hives, again, so that we're not comparing apples to oranges here if different people are using different genetic stocks. And again, we have beekeeping notes that address these different aspects. Okay, finally, submitting your data. This won't work unless we pool our information. All right, so if we all abide by that same experimental design, oh, sorry, go ahead. Jim, you have a question? What if you're using local stock and you're unsure of the genetic type? I believe that that is one of the options. And there are going to be fields in the online form or a place for you to write in on the written form, kind of explaining that. I know that it's very popular for people to catch feral bees, feral swarms, and that they're putatively Varroa resistant. This would be a great way to test that. And so you can do that. That it's not a commercial stock necessarily, but if it is a certain genetics that you would like to test, that is perfectly reasonable too. It's just make sure then that your control colony, the thing that you're not doing anything to, is your standard Italian stock. That's kind of the generic stock that most beekeepers out there are using. So use that as your control, and then use whatever genetic stock that you favor, use that as your treatment group. Does that make sense? Okay. All right. So again, on this website, the Swarm Collective, which is off of our entomology cooperative extension portal, it goes through this same experimental design and protocol. There's also a PDF that you can download that explains it and also provides a form where you can write down your data as you go through the next couple months measuring your mites, okay? And as I said before, there are also tutorial videos and other things there. Now, if you, once at the end of September, after you measure your mites for the last time, that's when you can submit your data. And so we've created, this is really the first time I've ever done this, a Google form, which is just an online form, the link of which can be found on that website, where it just makes it really, really easy to go through various pull-down menus and open text boxes and those kind of things, where you can just simply fill out these forms and enter in all of those data. So it'll just give kind of the state and county where you're at, how your colonies were established, all of those types of information that we just went over. The 4 different measurement periods, 2 before and 2 after your mite control strategy, and then enter in those mite levels, and then hit submit. And then that will then go into a database that we will then collect and analyze, again, keeping the name of the beekeeper anonymous. In fact, I don't even think that it asks for it. And then we'll be able to pool our data and analyze it with all these different factors to see, kind of what's working and what isn't. If you don't like online forums, or you hate Google, or you just don't want to submit it that way, you can certainly submit either via email or by sending it in by snail mail, the data sheet that is in the handout that is on the website, where again, it just asks these various questions for hive number 1 and hive number 2, and you just enter in the dates and the mite levels, and answer the rest of these questions, and then just send it in to us, and then we'll type it into that same data sheet so that we'll be able to capture your data as well. I should mention that if you have more than 2 hives, which I'm sure many of you do, and you want to try different things, or you want to do multiple replicates of the same thing, you can do multiple pairs. So you can do 5 pairs. If you have 10 colonies and you want half of them where you're not going to do anything and the other half where you're going to implement your mite control strategy, you can do 5 pairs if you wish. Just submit the data, each individual pair, and then we'll treat them separately accordingly. So I think it's presented in a fairly easy way, but there's going to be a lot of data that come out of this. Especially the more people we have, the more insights that we're going to have into any one mite control strategy, especially for some of the things that a lot of people aren't using. Lisa, you have a question? Yeah, sorry, I moved the hand outside to get it out of the way. So yeah, so this is just a table here that, again, you can download, and each one of these cells you can fill in the appropriate information. It's probably not easy enough to read here. It's just asking what dates do you put the sticky boards in, what date did you remove it, what's the total sticky board count, what's your sugar shake count, those types of things. You just fill out the information, you can send it in to us. Trying to make this fairly easy and fairly self-explanatory, but of course if you have any questions, don't hesitate to ask. So then when it comes down to it, analysis and reporting. So again, the more people we get, the more robust and the more informative the data are going to be. In essence, what we're going to do is we're going to take the number of mites in your control colony and subtract the number of mites in your treatment colony or your mite control colony. And so the larger that difference, the more effective that mite control strategy is, right? If the difference is 0 or even negative, then that means your mite control strategy is not working. But if the bigger it is, the better that it's working. And so as we average across different beekeepers that are doing the same thing but in different places, we can see on average what the average efficacy of these different mite control strategies are. And in doing so, we can also look to see how effective the screen bottom boards are, these different stalks are, or all of these different aspects. We're going to be able to look at them simultaneously. What we'll do then is we will post this information in our Wolfpack Waggle newsletter. We'll also hopefully share it on our Bees Network, the Beekeeper Education and Engagement System, because this is right up the alley of that philosophy of working together and learning from each other, and also hopefully publish it in a beekeeping periodical or trade magazine, so again that others can learn what we find. So be part of the research team. I think that, again, we have a really vibrant, active beekeeping community here. This is, as an active beekeeper, this is something that you're probably already doing in order to keep your hives healthy, to keep Varroa mites in check. There's lots of different options for Varroa control, and so let's share those data that you're already collecting. Let's Let's consolidate them in a very systematic way, in a very easy way, where all you're doing is taking 20 measurements on 2 hives. Send that in, and we can pool the data and analyze them together. Again, this is the link to the website right here, which again can be found off of our main main website as well. So with that, I also, before we end, want to remind you that our next webinar, we tend to intersperse these between the NCSBA meetings and other activities throughout the year. So our next webinar will be hosted by the Durham County Beekeepers, one of our newer chapters in the state of North Carolina, in the middle of September, September 15th. The topic hasn't yet been determined, but hopefully we will see you then. And with that, again, I would be happy to entertain any questions that you might have. I see that some people said that they couldn't see the table, so my apologies on that. I'm not entirely sure why that came through. But again, if you go to the website, you should be able to download that PDF and see the data table that will really facilitate and help you keep track of those numbers and be able to send them in when the time comes. Cheryl, you have a question? So the next webinar, again, this is on our website. We have them posted out. I think these are booked until January 2015, or no, June 2015, where we'll have one in September 15th, by the Durham County Beekeepers. I believe January 2015 will be the Chatham County Beekeepers, who will be virtually hosting us, and then June, this time next year, will be the Johnson County Beekeepers. So stay tuned to our website and the links and all the other information through your cooperative extension office and your county chapter. The question is, what is the number of Varroa mites that is acceptable? That's an excellent question. If you have answers to those questions, again, are found in the literature, found on our Bees Network, on the Varroa mite course that you can take online. It goes into that in detail. In essence, the sticky board count varies in what the threshold is when you really need to take action, because higher numbers with higher colonies is probably more acceptable than higher numbers at smaller colonies, right? So the range is anywhere between 60 and 180 mites per 24 hours, okay? So in essence, if you have more than 100 mites on a good-sized hive at the middle of summer or at the end of the summer, then you'll probably need to do something. But below that, if you only have 50 or 20 mites on a sticky board every 24 hours, then you probably don't need to actually take any action. So that's why the sticky board can be hard to interpret sometimes. And that's why I like the sugar shake, because it gives that percentage. And so in essence, the percentage for the number of mites per bee, the acceptable level is like 5%. So if you have 5 mites for every 100 bees, then that's probably too high a level, and your colony is probably going to suffer or get deleterious effects from that. So again, that depends on the time of year and other factors, but during this time, that's your treatment threshold. But again, we don't want you to treat when it reaches the threshold. We want you to treat during that time in between your 2 measurements. So this is actually kind of going counter to what we normally say, which is really only treat when it's necessary. But here we're actually asking you to treat at some point during August. And if you already have low levels, then you're not going to be able to knock them down very, very far, because they're already low. But it's still the comparison between your control versus your treatment hive that's going to be informative. Okay. But at the end of September, if your control colony, the one that you didn't do anything to, is higher than those thresholds, then I would very strongly encourage you to take some sort of action in that control colony also after the experiment is concluded. The Mecklenburg group here asks a very good question. That is, how much time should elapse between the sugar shake and the sticky board count? I don't think I mentioned this here, but I think it's mentioned in the little downloadable write-up. What we want you to do is put the sticky board in and then take it out 72 hours later and then conduct the sugar shake. Okay? It's best if you don't. And this is true for any time that you're using a sticky board, you really don't want to be going into the colony and being too disruptive to the colony while the sticky board is there, because the more activity and the more moving around of frames and everything you do, that can artificially inflate the number of mites that drop off, which is a good thing in the sense that it's killing off mites, but it's a bad thing because it's not accurately reflecting the mite load. It's inflating it, and it's giving you a higher count than you would if they were unmolested during that time. Okay, so what I would suggest is putting the sticky board in, waiting 72 hours, pulling the sticky board out, and then immediately going in and doing your sugar shake and taking it right then and there. Jim, you have a question? Requeening as an option for control. Absolutely. It is caging the queen, requeening, in other words, breaking the brood cycle, right? So if you can have a hiatus in egg laying, and therefore there's kind of a gap in the appropriate age of brood for the mites to hop into and reparasitize to complete their life cycle, that is a very effective mite control strategy that I believe is included in the pull-down menu on the Google form. But if it's not, there's a possibility to write that in as well. And so if you are doing that, that is absolutely a perfectly acceptable way to control for Varroa, and I think it's a pretty effective strategy too, and I think that's one mechanism by which, like the Russian bees and some of the others, can be effective as well, is their queens kind of naturally shut down during the summer dearth, which aids in that mite control trait that they have. And so definitely, if that's your control option, include it. That can definitely be captured here. Should we record the number of hive boxes, height, and number of frames of boxes? Yes, that is actually on the form. That it says, you know, how many brood boxes do you have and how many honey supers do you have. What I'd really like is the number of frames of bees and the number of frames of brood. But again, I think that starts to get a bit onerous, and of course that's changing over the course of the 2 months of the study and everything like that. So I just decided to keep it simple here, and we'll standardize by size based on how many boxes, and that is how many brood boxes and how many honey supers. And so that will be part of that, and we can help standardize for that. Yes, absolutely. Yeah, Paul, go ahead. Unless, Jim, you had a second question too. And you can use your microphone as well if you don't want to type or you're not that fast a typer. Yeah, so I'm recording it right now, and I will be posting this on our website, so you can get to it right from here. And it will have a link where it will go to the recording rather than the live session, since it will be moot at that point. And so you can send the link and send it along to anybody else who might be interested. We want as many people participating in this as possible, because the more the better. Well, if there are no other questions, I'm going to end the recording. And again, thank the Mecklenburg County beekeepers for being our hosts tonight. And thank you again.